Hi, everybody, and welcome to this week's lecture. What we're going to be looking at today is going to be how gene expression is going to be regulated. So what we're going to try to do is we're going to try to see if we can describe how gene expression is regulated. So exactly what we're doing with those genes and how we're going to be modulating or adjusting the amounts of genes that are going to be expressed or the level of expression or whether or not we're going to keep them on or off. So before we can even really talk about that, one of the first things we need to look at or kind of question is how and what does that really mean? So what I mean by that is we've been studying DNA replication. We know that each one of our cells has DNA in it within the nucleus, and we know that each cell, which has our DNA, contains everything we need in order to produce all the proteins and everything. That's why it's known as a control center, because again, it has everything we need in order to make the proteins that we need in order to facilitate life. Now, one of the big questions, or the two questions that we're going to look at, are going to be one, how do all of your cells in your body contain the same DNA? We're going to go ahead and explain that. And then how can you test your hypothesis? So the reason why we kind of bring this up is if all of our cells have the same DNA, does that mean all our cells are the same? And we know that that's not the case. And the reason why is because you can see we have all of these very specific specialized cells. You can see we have our blood cells, which are going to be very, very good at transporting oxygen and nutrients throughout the entire body. But then if we compare these to, let's say, our nerve cells, you can see that they are going to look drastically different. Our nerve cells, again, are going to be very good at sending signals in our brain and information. You can see our nerve cells, again, are going to be completely different from our heart muscles cells, which are going to be very good at contracting and pumping blood throughout our body. And you can see that these are going to be different as well compared to our small intestine cells, which are going to be great for absorbing nutrients and things like that. So again, each one of these has the DNA for our entire body, yet everything is completely different. Again, our blood, our nerve, our heart, and our small intestines all look drastically different and all have drastic different functions they're responsible for within our body. So how is this possible and what's going on with that? So again, we do know that all the cells of your body contain the same DNA. And we know that because when we start off with our fertilized egg, remember we're taking our sperm and egg cells, and then they're going to start dividing out. When they start dividing out, we're going to have more and more cells, and eventually we're going to go ahead and create an organism or a person. So we do know everything does have the same DNA, and we know this because of our cell division. And then how can we test that? Again, when we look into it, we can see everything has that DNA. But now we're going to move on to our next part, which is going to answer that second question, which is how can we have specialized cells? How can they all be different despite having all the same DNA? Now, this is going to be because of something known as gene regulation. When it comes to gene regulation, basically, just like the name suggests, it's going to regulate the genes. What this is going to do is either going to turn on or express a gene, or it's going to turn off or not express it. Basically, what that means is, again, our DNA has all of our instructions, all of our things we need in order to know how to produce our proteins, which again will do specific things within the body. Because we have all the instructions, again, we don't always need to read it all at once. If we need to build a specific thing like liver cells, we're going to look at that specific liver cell spot. So we're going to turn that portion on, but we're going to keep everything else off. And this is how we're going to get different cell types that are going to be uniquely specialized to do their jobs. So right here, you can see we have our gene, and whenever they are turned on, or again, they're going to be expressed, that means that that part is going to be red, and we can go and produce our mRNA, which again, remember, is going to go into our translation process, and it's going to go and produce our proteins. When our gene is turned off, right here, or again, not expressed, you can see that no mRNA here is going to be produced. So that means just nothing's going to be made, no message is going to be sent, and no proteins are going to be produced. So again, although all of our cells have our DNA, which again is going to be the blueprint for our entire body, only certain parts are going to be turned on or expressed, and certain parts are going to be turned off or not expressed. Again, we're only turning on the parts that we need in order to produce whatever we need in order to have those cells become uniquely specialized in order to do their job. Because again, we have so many different moving parts within our body. We have so many different cells that need to do very specific things. And again, it can only do that when it has the correct blueprints and it knows exactly what it needs to make in order for those cells to be able to do their job. Again, we don't want everything to be made all at once, all the time, because again, we only need specific things at specific portions in order to, again, make everything work in conjunction with each other so we can facilitate life. So let's go and take a look at an example of this and exactly what that means when we're talking about gene expression. What we have here is we have a liver cell and we also have a neuron over here. Now, both of these, again, are going to contain all the DNA for our body, but you'll notice that it is going to turn on and turn off specific portions that are going to be pertinent to its specific job. So we know that liver cells are going to be very good at breaking things down, removing toxins from the body, because again, it's going to be like a big filter to help us stay clean, maintain our homeostasis, and again, make sure that our lives are being run the way that it needs to be run. So you'll notice that right here, again, right in the nucleus, we have all of our DNA. And you'll notice that it has something known as our alcohol dehydrogenase. Now, what that is this is going to be enzyme that's going to break down alcohol into non-toxic molecules. Now, this is going to be incredibly important because, again, liver cells, they're going to be all about filtering, all about making sure that we clean everything up. So these alcohol dehydrogenases are going to be very important in order to help us break down alcohol into non-toxic molecules. So again, we can go ahead and pass those. So you can see that we have our DNA here, and you can see we have our first section, and then we have our second section here. You can see in our first section, we have our alcohol dehydrogenase. So this right here is going to be expressed or turned on because this is what we want. We want our body to produce these enzymes in our liver so that we can go and break down that alcohol into our non-toxic molecules. So again, this is very specific to our liver cells because again, this is exactly where we want these alcohol dehydrogenases. This is going to be the best place for them in order to do their job. Now you'll see that also on our DNA, we have our neurotransmitter genes. Now this right here is going to be very good at sending signals. They're going to be great at communicating, but we do not need these because again, the main job of the liver cell is to filter or clean things up. So this is going to be off. So that means that we are not going to produce any of these proteins here because again, we do not need to send any signals. We need our alcohol dehydrogenase enzyme in order to clean up our body. Now, it's going to be the exact opposite when it comes to our neuron. So again, neurons are going to be very important because again, they're going to be sending signals throughout the entire body. Now, what we have is a protein that is going to be known as a neurotransmitter. So a neurotransmitter is going to be very, very good, again, at sending signals. Now, you can see that we have our first position on our DNA, and we have our second position. And you can see we still have our alcohol dehydrogenase, because again, in our nucleus, we have our DNA, which contains all the information for our body. Now, since we're dealing with our neurons, which again are all about sending signals through our brain, we do not need our alcohol dehydrogenase. So this is going to be turned off. And again, the reason why is because this is great at, again, breaking down alcohol, but because our neurons aren't supposed to do that, that's not their job. That's what they didn't specialize in. They specialize in sending signals. And they do that by using neurotransmitters. So that means on our position on our DNA here, that's going to be turned on. So then we are going to start producing our RNA here in order to produce those proteins. So again, this is a great example of showing our gene expression. Although both of these have our nucleus with all of our DNA, only specific parts of that DNA are going to be utilized and read in order to produce what that specific cell is going to need. And by doing that, it's going to specialize that cell and it's going to allow that cell to do a very specific job, but do that job very, very well. So again, just a quick recap, our liver cells, they're great at filtering. So they're going to need an enzyme known as alcohol dehydrogenase because it's going to break down alcohol into our non-toxic molecules. It's going to do that by looking at our DNA. It's going to find the alcohol dehydrogenase portion. It's going to go ahead and read that, go ahead and make our RNA so we can start producing those enzymes. We do not need neurotransmitters in this area, so we are not going to be using this. So that is going to be turned off, meaning that this part is not going to produce any RNA. So it is not going to send that message to produce that enzyme. On the other end, our neurons do need our neurotransmitters. Those are going to be important proteins because they're going to be used to send signals. So on that same piece of DNA, because again, different cell, but we still have all of our DNA in our nucleus, it's going to go ahead and look at their alcohol dehydrogenase gene. We do not need that. That's going to be turned off because again, our neuron is specific to sending signals. It has nothing to do with filtering. So what it's going to do, it's going to find the second gene, our neurotransmitter. It's going to go and turn that on, meaning that it is going to go ahead and be read. Our RNA is going to be produced, and then it's going to go ahead and start producing those neurotransmitters. All right, so another thing I need to kind of mention when it comes to regulation of gene expression is gene expression isn't always going to be a specific on or off. Sometimes we can have it kind of in the middle. We might have a lower level, a medium level, things like that. The reason why is because this is going to allow us to precisely express which gene and how much we need of those. So again, our body contains thousands of genes, many of which can be turned on and off precisely and in the right places in order to produce whatever we need to produce. Now, when it comes to gene expression, genes can be thought of more of a modular dimmer switch, meaning that we can modulate or can have a specific amount. So again, when it's off, that means that nothing is being produced, right? Off, just like a light switch, if you turn it completely off, there's no light, no electricity going to the light bulb, turning it on. When you turn it on, we can have a very high level of mRNA being produced. But again, remember, we can kind of modulate the amount of it turning on and off. So we can kind of underexpress, we can overexpress, again, depending on that. And we can have different levels of mRNA being produced. Because again, certain parts of our body might need some of this being produced, other parts might need a lot more of it being produced. So again, it's just an idea of exactly how that gene expression is going to work and how we can, again, adjust the amount that's going to be expressed, which directly affects how much protein is being produced. So again, think about gene expression kind of like a dimmer switch. Although we can completely turn it off and nothing will be produced, or we can turn it on and we can have high levels being produced, we can also modulate it in between here to have the specific or precise amount of gene expression we want in order to have whatever we need to be made in that specific spot. All right. And that is going to go ahead and conclude our lecture for this week. So what I'd like you to do is to go ahead and take about 5 seconds, go ahead and pause this, see if we can answer these 3 questions, and then we'll go ahead and check them out together as a class. All right, welcome back. And these are going to be the answers to those questions. So as always, if you have any questions, comments, concerns, please feel free to reach out. And I look forward to seeing everybody in the next lecture.